Hi everyone. In the previous part of microbial metabolism, we had gone through the introduction about the respiration, and depending upon the type of electron acceptor, how the respiration is going to be classified into three types, that is aerobic respiration, anaerobic respiration, and then fermentation, especially in microorganisms. Okay, so in this part we are going to discuss about the aerobic respiration, which is going to occur in the three stages, that is glycolysis, citric acid cycle, and electron transport chain. So let's see what is an aerobic respiration. So when molecular oxygen is the terminal electron acceptor, that means oxygen is going to act as a terminal electron acceptor, then that metabolic pathway is going to be known as aerobic respiration. In this aerobic respiration, a molecular process by which cell is going to consume the oxygen and produces the carbon dioxide, water, and as well as the ATP, nothing but the energy is also being generated, isn't it? So this is what happening in the aerobic respiration. This also I gave a brief introduction in the faster part, that is microbial metabolism. Now, this aerobic respiration is going to occur in three stages. That means one molecule of glucose is going to be undergoing the oxidation process by having the three stages, that is glycolysis, citric acid cycle, then electron transport chain. So first of all, we will discuss about the glycolysis. Then in another part, we'll discuss about the citric acid cycle. And then in some other part, we'll discuss about the electron transport chain. So let's begin what is happening in the first stage, then what's happening in the second stage and the third stage. In the first stage, one molecule of the glucose is going to be converted into the pyruvate or pyruvic acid. Okay, 3-carbon compound. Now, from the first stage of entering into the second stage, that 3-carbon compound have to be converted into a 2-carbon compound called as acetyl-CoA enzyme or acetyl-CoA. Now, this acetyl-CoA will combine with the oxaloacetic acid or acetate of TCA cycle, and that's how it starts the second stage called as TCA cycle. And whatever the reduced electron carriers or reduced coenzymes are going to be there, so this all enters into the electron transport chain and ultimately gives the ATP, that is energy source, okay, which we call it as oxidative phosphorylation. We will discuss all these things in detail one by one. Now let's begin with the first one, glycolysis. What is a glycolysis? So the glycolysis is going to be considered as the first phase of our aerobic respiration. So what's happening in this glycolysis? So in this glycolysis, the organic molecule, that is our glucose, is partially oxidized to smaller molecules. That's why we call it as a catabolic reaction, isn't it? Usually with the generation of ATP and some sort of reduced coenzymes like NADH. So if a substrate molecule is glucose, it's initially break down to the pyruvate molecules. And especially coming to the microorganisms are going to employ several metabolic pathways to catabolize the glucose and other sugars. And there are three important routes of breakdown of this glucose to pyruvate. So what are there? There are EMP pathway, ED pathway, and HMP pathway. EMP, you know very well what is the EMP pathway. In general, we call it as glycolysis commonly, but there is a difference between these things. Embedded Mehrhoff-Parnas pathway, and then the ED pathway, enter-Diotroff pathway, which we specially seen only in the bacteria, some bacteria. Then HMP pathway, hexose monophosphate pathway. So we will discuss all these three routes of breakdown of glucose into pyruvate in different parts. So let's begin with the first one, that is EMP pathway. So as we know, this EMP pathway, the sequence of reactions which convert glucose to pyruvic acid along with the production of ATP is known as glycolysis. What is the glycolysis? The sequence of reactions which convert glucose to pyruvic acid along with the production of ATP is known as glycolysis. And most common pathway in glycolysis is the EMP pathway, that what we observe in the animal cells or in the plant cells is the same one, that is EMP pathway. And the EMP full form, as I told you, is the embedded Payoff pathway, which is also known as glycolytic pathway or hexose biphosphate pathway. Remember this name because we'll discuss about the hexose monophosphate pathway also, HMP pathway. So the EMP pathway is also called as hexose biphosphate pathway, which is the most common pathway for glucose degradation to pyruvate. Okay, so this EMP pathway, as I told, is going to be found in all major groups of microorganisms and functions both in the presence or absence of oxygen. This is once again important. This EMP pathway is going to function both in the presence as well as in the absence of oxygen. That is, it is common to all aerobic respiring organisms and to most aerobic as well as the fermentative bacteria, except, so here it's an anaerobic, it's not aerobic. Most anaerobic and as well as the fermentative bacteria, except one bacteria, that is archaebacteria, which do not use this pathway. So it is going to use some other pathway, not this EMP pathway. And then let's see the reactions that are occurring in the EMP pathway. Then we will move to the next type. So what are the reactions occurring? Glycolysis, as I told you, is a breakdown of the glucose up to the formation of pyruvic acid. Until the glucose is going to be converted into pyruvic acid, this is going to continuously break down. And each glucose molecule is going to form two molecules of pyruvic acid. Remember this one. One glucose molecule gives rise to how many pyruvic acid molecules? Two. And the whole process of the glycolysis is going to occur in 10 steps. Okay, whereas in the TCA cycle it will be 8. Okay, just for remembrance I'm telling you, the whole process of the glycolysis is going to occur in the 10 steps. Out of the first 5 steps are going to be called as preparative phase, and the second, that is the other 5 steps, are going to be called as payoff phase. One is the preparative phase and second one is the payoff phase. And this, for example, if you want to cook, you are spending energy. After cooking, if you eat, you are going to get the energy. So that's how preparative phase, payoff phase. So EMP pathway initiates by coupling the conversion of ATP to ADP, or in the case of bacterial cells, by using some sort of a special system called as PEP-PTS system, that is phosphoenol pyruvate phosphotransferase system, is directly getting converted into glucose 6-phosphate during the transport across the cytoplasmic membrane or plasma membrane. And the energy balance of this PEP-PTS system or this EMP pathway is roughly equivalent to that of direct conversion of glucose to the glucose 6-phosphate using ATP. So overall, just remember that we are going to have the 10 steps in the glycolysis, and the first 5 steps are going to be called as preparative phase, and the second, that is the next 5 steps, are going to be considered as payoff phase. So this is all about the first 5 steps, and then coming to the next 5 steps. These are the 5. So totally altogether we have 10 steps. So this I will represent it in the form of a flow chart, then you can understand. Let's come to the first step, that is the conversion of glucose into glucose phosphate. This step is going to be named as phosphorylation of glucose. So that's how here is phosphorylation of glucose. So what is happening here? The activation of the glucose, that means it have to give some energy, so that is ATP. So the activation of glucose takes place by a reaction called oxidative phosphorylation, and in this reaction a phosphate group is attached to glucose by a low energy phosphate bond and converted into glucose 6-phosphate. And this whole process is going to occur in the presence of an enzyme called as hexokinase. And this hexokinase requires some sort of cofactors called as magnesium ions for its activity. and the phosphate group is derived. So where did this phosphate is going to come? From the ATP molecule, which gets broke down into the form of ADP. So that's how the overall step is going to be of glucose plus ATP, giving rise to glucose 6-phosphate plus ADP in the presence of an enzyme called as hexokinase. So that's what here it is going to be shown like this. Then what is the second step? The first step is phosphorylation of glucose. Then what is the second step? The second step is isomerization of glucose 6-phosphate to fructose 6-phosphate. Okay, so this is going to be of, the name itself is saying it is isomerization. So obviously the enzyme involved here is isomerase. So what is happening here? The second reaction in the EMP pathway is isomerization of glucose 6-phosphate to the fructose 6-phosphate. And the reaction is catalyzed by phosphoglucose or phosphohexose isomerase, anything you can call it as. And this step is a reversible step. That means the fructose can be converted into glucose 6-phosphate, or the glucose 6-phosphate can be converted into fructose 6-phosphate. That's how we call this as a reversible step. So second step is isomerization. So we are going to move to the third step, that is where the second phosphorylation occurs. That means one more ATP is going to be involved in conversion of this fructose 6-phosphate into fructose 1,6-diphosphate or bisphosphate. So let's see this. So here is the step. So what is happening in this reaction? The fructose 6-phosphate is phosphorylated again to form fructose 1,6-biphosphate or diphosphate. And the whole reaction is catalyzed by an enzyme called as phosphofructokinase, and which again requires the magnesium ions for its activity. Okay, and it is a key enzyme in regulating the whole process of glycolysis. The phosphofructokinase enzyme is the key enzyme which is regulating the whole process of glycolysis. The high energy bond of ATP is utilized for the synthesis. The low energy ester phosphate bond of this 1,6 diphosphate, and that's how you are going to have the formation of fructose 1,6 bisphosphate. So altogether, the first reaction of conversion of glucose into glucose 6-phosphate, then the second reaction from 6-phosphate to fructose 6-phosphate, and then from the fructose 6-phosphate, it was converted into fructose 1,6 bisphosphate. So how many reactions are now? 1, 2, 3. We have finished 3. So I told you in the preparative phase we required 5, isn't it? So next, moving to the fourth reaction, that is cleavage of fructose 1,6 bisphosphate into two molecules. What are those? Let's see. So now this fructose 1,6 bisphosphate splits into two different 3-carbon atoms or molecules. That means fructose is a 6-carbon compound, and it is going to be cleaved into two equal parts of having the 3-carbon, and here also 3-carbon. So one is going to be dihydroxyacetone phosphate, simply called as DHAP, and glyceraldehyde 3-phosphate, which we call it as phosphoglyceraldehyde also, PGAL. And the enzyme here involved is going to be of our aldolase. So aldolase enzyme is going to be used to cleave the molecules into two. So that's how the fructose 1,6 bisphosphate is going to cleave the things into two, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So here you can see with the help of an enzyme called as aldolase. Now the next step is isomerization. Again, isomerization is a reversible reaction, isn't it? So where it can have both the interconversion. That means the glyceraldehyde 3-phosphate can be converted into dihydroxyacetone phosphate, and this can be converted into glyceraldehyde 3-phosphate. And the enzyme here is isomerase enzyme. So this is all about the first 5 steps of preparative phase. So 1, 2, 3, 4 and 5. Now the cooking is done. Now we have to get the energy, that is going to be of payoff phase. So here comes the 6th reaction, that is phosphorylation and oxidative dehydrogenation. Okay, so here you can see from here 6, 7, 8, 9, 10 are going to be the payoff phases. The phosphorylation and oxidative dehydrogenation is going to involve one, the ATP synthesis, and then removal of the hydrogens. So let's see what is happening here. So this in this step, as I told you, here you are going to have the payoff phase, which is also called as energy yielding phase. The glyceroldehyde 3-phosphate oxidizes to 1,3-bisphosphoglycerate, 1,3-bisphosphoglycerate with an enzyme obviously called as glyceroldehyde 3-phosphate substrate name, and the enzyme name is dehydrogenase. And during this reaction, that is conversion of glyceroldehyde 3-phosphate to 1,3-bisphosphoglycerate, NAD plus is going to be reduced, yielding NADH plus. And we know this NADH will enter into the electron transport chain and synthesizes the ATP by a mechanism called as oxidative phosphorylation, isn't it? So that's how we'll get the ATP indirectly, not directly. Okay. Next, the next step of payoff phase is 1,3-bisphosphoglycerate is going to be converted into 3-phosphoglycerate. That means here direct ATP generation is going to occur. If you see here, the high energy phosphate, that is in the biphosphate, 2 phosphate groups are there, and that is going to be given off, and that's how it became the 3-phosphoglycerate. The high energy phosphate group in this 1,3-bisphosphoglycerate is transferred to ADP, resulting in the formation of ATP. And that's how what we'll get finally. One is lost, so obviously 3-phosphoglycerate will be formed. And the enzyme that is catalyzing this reaction is phosphoglycerate kinase. Okay, what is it called as? Phosphoglycerate kinase. And one more important point here is the formation of ATP is going to occur because of a substrate is involved. So we call this as substrate level phosphorylation. Substrate level phosphorylation, because uh, a phosphate, that is high energy phosphate, is going to be transferred from a substrate molecule. So that's why we call this as substrate level phosphorylation. We will discuss in further videos. Okay, so just remember, here it is going to be the oxidative phosphorylation and here it is substrate phosphorylation. Just remember the names. Then we got now 3-phosphoglycerate. Now this 3-phosphoglycerate is going to be of, again going into the isomerization step, giving rise to 2-phosphoglycerate. That means the phosphate position is going to be moved from the third carbon to the second carbon. So that's how it becomes a phosphoglycerate or 2-phosphoglycerate. Okay. Now this enzyme that is involved here is going to be the mutase. In this reaction, as I told you, phosphoglycerate mutase catalyzes a reversible shift. So here you can see how this is reversible shift of the phosphoryl group between the 3-phosphoglycerate and the 2-phosphoglycerate. Then the next step is going to be of 2-phosphoglycerate. So where the dehydration is going to occur. See here, the removal of water is going to be occur in the presence of an enzyme called enolase. Okay. On dehydration, that is a removal of water molecule, 2-phosphoglycerate gets converted into phosphoenol pyruvate. It is getting converted into phosphoenol pyruvate. And now finally, this phosphoenol pyruvate is getting converted into the pyruvate form with the help of an enzyme called as pyruvate kinase. So this is how we are going to have totally 10 steps of glycolysis. The first 5 steps up to here, it is going to be called as preparative phase, and the next 5 steps are going to be called as payoff phase. So these 5 are going to be of payoff phase and these first 5 are going to be of preparative phase. And then coming to the overall reaction of glycolysis, the glucose is going to have the utilization of the 2 ADP molecules, and 2 NADP plus molecules and giving rise to 2 pyruvate and 1 NADH molecule and 2 protons. Sorry, here it's a 2 NADH and 2 protons and 2 ATP. How you get 1 glucose but here 2? Because one glyceroldehyde is giving rise to one NADH, two ATPs, isn't it? Now this dihydroxyacetone again enters, converts into glyceroldehyde, and that will enter. So altogether we are going to have the two molecules of pyruvate synthesis, four ATP molecules, out of which two are utilized, so we are going to minus them, and we are having that 2 NADH. Okay, so here is 2, here is 2, here is 2, here is 2. So overall how many pyruvates were synthesized? 2 were synthesized, isn't it? So remember, from one glucose molecule we had the synthesis of 2 pyruvate molecules, 2 NADH, 2 ATP molecules. Now coming to the significance of this glycolytic pathway or glycolysis, as we know, this not only serves not only to oxidize the carbohydrate to pyruvate, and it is also used to phosphorylate ADP, but also provides precursors for many other pathways like glucose 6 phosphate is a precursor going to be for polysaccharide, pentophosphate, and aromatic amino acids too, isn't it? And especially coming to the HME pathway. Okay, then fructose 6 phosphate is a precursor to many amino sugars, and then dihydroxyacetone phosphate, that is DHAP, is going to be acting as a precursor to phospholipid synthesis and to some of the amino acids like glycine, serine, and cysteine. Even the last compound, last but one, that is phosphoenol pyruvate, is also acting as a precursor to aromatic amino acid synthesis and to some sort of a lactile portion of muramic acid too. So these all going to, that means whatever the metabolites that are present in the glycolysis are all going to have some sort of a importance in different pathways or in different synthesis. So these are all the significances of our glycolytic pathway or glycolysis. So this is how we finish the first uh, type of the breakdown of the glucose molecule, that is EMP pathway. Then we will move to the second type, ED pathway, in the next part. Thank you.